So welcome back, everybody. We're going to continue discussing the mollusks. Last lecture, we looked at the gastropods, such as snails. So the first group or class we're going to look at today are the bivalves. There's close to 30,000 species, and they're the second largest group. And it includes animals such as clams, oysters, mussels, and scallops. They have a sheet-like mantle consisting of two valves, or shells, and it covers these laterally compressed animals. Most are filter feeders, and they even have the ability to remove some bacteria from polluted water. We're now going to discuss the shell and associated structures. The shell actually is composed of two convex halves, and they're called valves. The shell is actually a single structure, including the two valves. Along the dorsal margin of the shell is a proteinaceous hinge and a series of tongues and grooves, or little teeth in a groove. And they're modifications of the shell, and so we can call them teeth. And that's important because with the hinge and the teeth in the groove, it prevents the shell from twisting when they close. The oldest part of the shell is the umbo, which is a swollen area near the shell's anterior margin, just where the hinge is. So this is a diagram of a bivalve that's open, and we can see the two valves. We can also see the line of the mantle attachment on both of the valves. We can also see the adductor muscle scars that close the two valves. In this area here is the dorsal side of the animal, or top of the animal, near the hinge. And away from that is the ventral side of the bivalve. As mentioned, we have a hinge between the two valves, and it's kind of elastic, rubbery. And along with the teeth, when it's closed, the teeth fit into the grooves on the other side of the valve, and it prevents the two valves from slipping past each other. So they're kind of locked. And also in the diagram, you can see the umbo, which is the oldest part of the shell. The shell is continuous along its dorsal margin, but the mantle in the region of the hinge secretes more protein than calcium. Thus, it results in an elastic hinge, and it's the elasticity that allows the two valves of the shell to open when the muscles are relaxed. And as just indicated on the former slide, the adductor muscles at either end of the dorsal half of the shell, when they contract, they close the shell. So this is another diagram looking at some of the features of a bivalve. And so we can see on the outside of one of the valves, this is the umbo, the oldest part of the shell. There's the foot, and the foot usually sticks out of the valves at the anterior end. And at the other end, the posterior end, we have the incurrent siphon and the excurrent siphon. And here's a shot of the two valves and the elastic hinge. So when the adductor muscles are relaxed, the two valves open. On the lower part of this diagram, we can see the imprint of some of the features. For example, the anterior adductor muscle, posterior, where the hinge would be. This is the location of the siphons. Not all bivalves have distinct siphons or tubes coming out. And this would be the insertion area where the mantle would fit in. And on the lower right, we can see when the valves are closed, the adductor muscle have contracted, and the two valves are now shut. So the mantle attaches to the inside of the valves of the shell, and they're around the adductor muscles and near the shell margin. And as we mentioned in last lecture, if a sand grain or parasite lodges between the shell and the mantle actually secretes nacre around the irritant and can form even a pearl. We saw this diagram last lecture, and perhaps a grain of sand is lodged between the shell and the mantle, and it lays down nacre and forms a pearl. So we can make artificial pearls with oysters, and there's also natural pearls. So we're going to talk a little bit about gas exchange, a little bit about filter feeding and digestion. First off, bivalves don't move around very much, so they're basically sedentary. And they're filter feeders. And because they don't move very much, they don't have a very distinct head. But they do have a radula, and except for very few species, very prominent gills have cilia on them. And when we actually look at the gills, in the previous slide, we saw the foot sticking out between the two valves at the interior end. And inside the shell, the gills look like little leaves on a two pair on either side of the foot. So the gills are full of cheeks with one end attached to the foot and the other end attached to the top of the mantle. And there's a mantle cavity ventral to the gills where water comes in at the posterior end, and that's called the inhalant region. And the cavity dorsal to the gills at the posterior end is the exhalant region. So this is a diagram actually looking at the development of the gills, and I'm more interested in the bottom diagram. And so this is the two valves. The pinkish area is the mantle. There's the foot, and there's two leaf-like gills on either side of the tongue, on the inside of the valves. And you also kind of saw that in the lab when we dissected a clam. So here's a really detailed shot of what I just mentioned. So a lot of detail here. Let's start with the shell. This side has been removed. There's the mantle. There's the foot. And as mentioned, there's two leaf-like paired gills on either side of the foot going from the back to the front. And they're important because they get oxygen, but they also filter particulate matter. So at the posterior end, we have an incurrent siphon or aperture where water comes in, is moved along the gills and goes through some little pores on the side, up water tubes, and then merge to a chamber at the top, and then leave through the excurrent aperture, or it could be a siphon in some species. So again, water comes in, goes through these little holes on the gills, goes to a large tube at the top of the gills, and out through the back of the excurrent aperture. And I should mention that there's cilia on the gills, and they also produce mucus. So particulate matter gets trapped on the gills, and the mucus with the cilia move the particulate matter down to the bottom of the gills, and there's a little groove in the bottom, as you can see here. It's called a food groove. And the mucus and food get moved along towards the interior end of the animal via this little groove and cilia, and then go into the mouth at the interior end. So here's another shot of much the same. So again, we're looking at a small section of this side. We can see water going through the pores and out. Blood is also going through the gills, so it's getting some oxygen, getting rid of CO2. So it has a dual function. And nonetheless, it also filters particulate matter, and it moves it along the food grooves at the bottom of the gills to the mouth at the interior end. So you might be wondering the actual mechanism by which water is moved through the bivalve. And it's actually the cilia that move water into the mantle cavity through the incurrent opening, or it could be a siphon. So for species that hide under mud or sand, they have long tubes where they can bring water in while they're being hidden and get rid of that water. So water moves in through the incurrent opening via cilia. Water moves from the mantle cavity into small pores on the surface of the gills. And then there's little ventral channels in the gills, and the water moves up those channels, and we call them water tubes. And as the water moves through the water tubes, both blood and water are in close proximity, and gases are exchanged by simple diffusion. So the gills pick up oxygen and get rid of CO2. Water eventually exits through a part of the mantle cavity at the dorsal or top part of the gills, and we refer to that as the supabranchial chamber. And then that water exits through the excurrent opening in the mantle. As previously mentioned, the gills trap food particles. And once trapped, the cilia, along with a little bit of mucus, move them to the gill's ventral margin. And at the ventral margin, there's a little food groove. And the cilia, again, along with a little bit of mucus, move the particles along that ventral margin towards the mouth. And so this is a relatively detailed diagram of what's going on. So we're looking at this side of the shell, or the valve has been removed. We can see the foot. So this is the interior end, and this is the pair of gills. So water comes in through the gill pores, up the tubes, and then out through the excurrent aperture or opening. And as mentioned, particulate matter get trapped on the side of the gills in a little bit of mucus, and the cilia move them down to a little groove on the bottom of that gill called a food groove. And in that groove, the mucus and cilia move towards the mouth of the animal, the opening. And just on either side of the mouth are these little labial palps, look like small little leaves, and they sort the particulate matter. So if it's too big, they move it to the side and then get rid of it through the excurrent flow. So on the bottom right, we can see a little more detail. So this is the other pair on the other side of the foot. This is the mouth. These are the labial palps that I was talking about that sort the particulate matter. And this is the visceral mass, including the stomach, the intestines, the gonads, the heart, the kidneys. And so some of those are some of the structures within a bivalve that we've looked at previously. So the cilia also cover these two leaf-like labial palps on either side of the mouth, and they sort the filtered food. And as mentioned, the cilia carry small particles to the mouth, and the palps and the cilia move the larger particles to the edge of the gills, and they reject that material. And the rejected material is called pseudofeces, and it falls or is thrown onto the mantle. And a ciliary tract on the mantle also transports them posteriorly. And when water moves out of the valves, it moves those false feces out. So in many species, the water is forcefully moved out of the mantle cavity. The actual process of digestion in bivalves is pretty complex, although it is similar to other mollusks. So food entering the esophagus gets entangled in this mucoid food string, and it extends all the way to the stomach. And it's rotated by cilia lining the digestive tract. In the stomach, there's a crystalline style that projects into the stomach, and it comes from a little pouch called a diverticulum, and that little pouch is called the style sac. And enzymes that are used for carbohydrate and fat digestion are incorporated into the crystalline style. And in the style sac, there's cilia, and it rotates the style against a chitinized gastric shield. And this abrasion, along with the acidic conditions in the stomach, dislodges the enzymes. The mucoid string winds around the crystalline style as it rotates, and it pulls the food string further into the stomach from the esophagus. This action, along with the acidic pH in the stomach, dislodges food particles in the food string. And there's further sorting that separates fine particles from undigestible coarse material. The coarse material is sent to the intestines. The smaller material that is now partially digested moves from the stomach into digestive glands on the periphery of the stomach. And in there, they undergo intercellular digestion. Ultimately, the cilia carry the undigested waste from the digestive glands back to the stomach and then to the intestines. The intestines empty through the anus near the excurrent opening, and the excurrent water carries feces away from the inside of the bivalve. And so this is a diagram, a simplistic diagram looking at some of the structures in the digestive system. So we can see the foot, we can see the gills, pair of gills on the other side of the foot. We can see the mouth in this area, the labial palps. This is the esophagus, this is the stomach, and then the intestines. And this is the anus, and it empties near the excurrent aperture or opening at the posterior end of the bivalve. And within the stomach, we have this little style sac and this crystalline style. And there's cilia within this style sac that rotate this crystalline style. And so it pulls this mucoid string into the stomach as it wraps around this rotating style. And in the stomach, it undergoes digestion. And so undigestible particulate matter moves through the intestine thereafter, whereas food particles move into little pouches around the stomach for further digestion. So this slide looks at some of the details involved with the esophagus, the stomach, the style sac, and the intestine. So this is the inside of the stomach, and this is the style sac with cilia lining it. This is the crystalline style that is rotated by the cilia, and it pulls in the mucus food cord or string, and so it rotates and pulls it along the esophagus from the mouth. And it's kind of like you using a fork to twist a strand of spaghetti around it. So in the stomach, enzymes mesh with the mucus cord or string. They start digesting carbohydrates and fats. The crystalline style rotates against this gastric shield, and it dislodges particulate matter. Large undigestible material is sorted in this area down here of the stomach, and so the undigestible material moves along the intestine. Smaller particulate matter that is partially digested moves into these little digestive glands on the outside of the stomach for further digestion. Let's look at some of the other organ systems and their functions. First off, blood flows from the heart to the tissues of the sinuses. It goes to the kidneys, to the gills, and then back to the heart. And the mantle lines the inside of the valves. And because it has a relatively large surface area, it probably provides an additional site for picking up oxygen in the blood. And in some species, there's actually a separate artery or aorta that delivers blood directly to the mantle. And also there's two kidneys below the pericardial cavity where the coelom is, and their ducts connect to the coelom at one end and then open at the nephridiopores in the interior region of the suprabranchial chamber. So this is a diagram looking at some of the structures involved in the circulatory system in a bivalve, and the heart is on the dorsal side of a bivalve within the coelom. And we have a number of vessels. In this diagram, they call it aorta, where blood goes to the front of the bivalve and to the back, and various vessels that take blood through structures such as the gills and other things. So in mollusks, we have a open circulatory system where blood goes to different tissues and organs. So for example, in this diagram, blood is delivered to the muscles in the foot, and it bathes those tissues of the muscles with blood, collects the blood, and delivers it ultimately back to the heart. So blood is not always confined to a conduit system, so it's an open circulatory system. As mentioned previously in this presentation, bivalves don't have a very distinct head region. And in fact, it's reflected in the nervous system. The nervous system in bivalves consists of 3 pairs of interconnected ganglia. And those ganglia are associated with the esophagus, the foot, and the posterior abductor muscle. When we look at sense organs, the margin of the mantle is the principal sense organ. It may have sensory tentacles on it, or it could even have photoreceptors. Scallops, for example, have photoreceptors on the margin of the mantle, and they actually form very complex eyes. They have a lens and a cornea, and they also have statocysts for equilibrium. So as mentioned in the previous slide, bivalves have 3 pairs of interconnected ganglia. So there's the pedal or foot ganglia. There's the cerebral ganglia. And at the back is the visceral ganglion. So it's reflective of their sedentary existence. So this is a picture of a scallop, and we can see on the edge of the mantle sensory tentacles, and there's also statocysts, but they actually have a number of complex eyes or photoreceptors with a lens and a cornea. And of course, scallops kind of swim through the water. They open and close their valves and squirt out water from the back and kind of swim through the water. So they need their photoreceptors to see essentially where they're going. We'll now start talking about reproduction and development. So most bivalves, there's a male individual and a female individual, although some have both gonads, male and female, in the visceral mass. Nonetheless, the gonads are kind of around the looped intestines, and the ducts from these gonads open directly into the mantle cavity, or perhaps by the nephritopore from the kidneys to the mantle cavity. Most species exhibit external fertilization, so sperm and egg exit through the suprabranchial chamber of the mantle cavity through the exhalant opening, and the sperm fertilizes the egg. It develops into a trochophore larva, into a veliger larva. Ultimately, this stage settles onto a substrate and transforms into the adult form. Most freshwater bivalves, however, brood their young. So fertilization actually occurs in the mantle cavity by sperm being brought in with the inhalant water. Some brood their young on the female gills, and they actually don't have a very visible trochophore or veliger stage. So they're much reduced. So in a lot of freshwater species, they have a reduced trochophore or veliger stage. And the young clams are actually shed from the gills. Some freshwater species brood their young through a modified veliger stage called a glochidium. And the glochidium is actually a parasite on fishes. So these little larvae, and they have 2 little valves, and on each valve they have little tooth-like hooks. And so they hang on to gill epithelium and feed on the host tissue. And in fact, some fish may form a cyst around the larva. The mantle of some freshwater species actually lure the fish to the bivalve, and the females squirt the little glochidia onto the fish and onto the gill epithelium. So again, the mantle cavity, or at least the mantle tissue of some freshwater species, have an elaborate modification that presents a fish-like lure to entice a predatory fish. So as soon as the fish gets close, the bivalve ejects the glochidia, which is the dispersal stage, onto the fish. And a few weeks later, after the larvae are developing, the little miniature clams fall from its hosts and take up their filter feeding lifestyle. So on the top is a little diagram of the structure of a glochidia. There's a tiny little valve on either side and a little tooth on the inside. And so this is a female bivalve, and part of the mantle is developed into what looks like a little fish-like lure. And it entices a fish close to this and thinking it's a meal. And when it gets close, it ejects all these little glochidia onto the fish, and a lot of them actually they close and hang on to the gill epithelium and feed and develop, and then they drop off and assume their filter feeding existence. So this is a close-up of the glochidia, and you actually saw this on the slide in the lab, and you actually drew it. And there's mom with the little fish-like lure, enticing a fish to come by. So before we finish with the bivalves, I'd like to mention something, and I talked about this when we first started this chapter, that in the old days they used to use the shells of various mollusks, especially bivalves, to make buttons for garments. And they're very durable. In those days, we didn't have plastics, so they used to drill out large diameter pieces of the shells and drill 2 little holes in each and then sew them onto garments. And I have a little story. When I first came to Fort Kent, I've lived on Shred Hill since coming here, and was walking through the woods with my dog, and I saw something shiny under the leaves, and I thought it was a coin. However, when I removed the leaves, I actually found a button with 2 small holes, and I talked to a historian, and they're pretty confident that it was made somewhere in the 1850s. So we used to use bivalve shells for making buttons for garments. So this is probably a good place to end. And next time we'll start with the cephalopods, talk a little bit about octopus, squid. And I think you'll find this group or this class very interesting. They do a lot of research on this group. And as always, until next time, please stay safe.